Hi all, Dr. Clark here again, and today we're going to continue with evolution and talk about the evidence for evolution. Some of this will be evidence that Darwin had when he came up with the theory of evolution by means of natural selection, but others will be advancements and evidence for evolution that has came with the advancements of technology, mainly genetics. And so we're going to approach that today, and we'll talk about the linkages between organisms and DNA, which has provided a lot of evidence for evolution. All right, to start with, Darwin had quite a few lines of evidence. The main one that he used was the fossil record. And the fossil record showed macroevolution, or evidence of macroevolution, which was controversial for most people. Large-scale changes, like going from birds, or going from dinosaurs to birds, or from reptiles to mammals, fish to amphibians. These large-scale changes, which we call macroevolution, was controversial during the time in which Darwin presented this material. But the fossil record was, at least right when he published the material, it was pretty incomplete. There wasn't very many transition species. So transitional forms like half amphibian, half fish, or half bird, half dinosaur, half mammal, half reptile, those were not really present in the fossil record. We now have them. We have transition fossils, and we'll talk about some of them. But during Darwin's time, paleontology was kind of just getting going, and that evidence was not really there for Darwin. But the evidence that was there were like remains of organisms, bones, tracks, fecal material, these kind of things that showed a great deal of diversity of organisms that once roamed the planet. And even, far before Darwin's time, geologists knew how fossils were created. They're created under intense pressure, and often that pressure is provided by things like water. So when an aquatic organism dies, it goes to the bottom of the lake or ocean, sediment covers it, and then that pressure from the water being on top of it eventually compresses that sediment and forms rock. Now, that alone is part of the reason why the fossil record is incomplete. If organisms fossilize, and you have to have the right conditions for a fossil to form, you have to have organisms that are going to die or be deposited pretty much in water, and that's the best way for fossils to occur. So if you have a lot of desert organisms, a lot of reptiles and things like that, there's not a very good fossil evidence of them because they just didn't get buried in water and they didn't get covered by sediment from the water. In some regions it happens, but this is why we have this huge amount of fossilized aquatic organisms and not very many fossilized terrestrial organisms. We have far more fish fossils than we have anything else, except for maybe snails and invertebrates and things like that. But it's because they live in a habitat that fossils readily form. The other thing that fossils give us the capability now with Madame Curie's work and then the people that followed her with the half-life of radioisotopes, it allows us the ability to date rocks or date fossils. And so using argon or some of these other elements that have large half-lives, million-year half-lives, 100-million-year half-lives, we have the ability to see when did certain organisms die, how long have they been dead, so how many millions of years ago was it that they got deposited or died on the planet. That's a big deal because it allows for us to build evolutionary trees and link groups together. The other thing that fossils really give evolutionary biologists the capability, or the evidence that's provided for by fossils, is evolutionary change. Because fossils can be treated as individual samples. So let's say you're looking at a mountainside and you have fossils at the base of the mountain. You know that those fossils are older, those organisms are older than the organisms above them, and all the way up the mountain. So therefore, if you have a series of organisms on the mountainside, you can then see examples of how did that organism change over the millions of years that it took for the organism to develop into either another organism or change body form and things like that. Because each one of those fossils can be treated as an independent data point, it really allows for us to independently date them, and we know that the fossil above them had no effect on them. So it's not that the fossil above them is inbreeding with them and causing these, these differences or anything like that. So that's one of the nice pieces of fossils, is that they're independent of one another. So then, like I said before, you can see successive changes. You can see changes throughout time, and you can link that to other data that you might have. And finally, this is probably our best source of visual macroevolution. So we can visually see the differences between one organism and another organism, or the similarities between the two, through fossil evidence. Okay. We can now do that a lot with genetics. Okay. In a different way, we can tweak genes and things like that, turn genes on, turn genes off, so we can get changes that we think probably had occurred, genes that probably mutated from one form to the next. For example, and we'll talk more about this, but you can take a chicken and you can turn the genes off that allow the chicken to produce feathers, and you just tweak the gene a little bit, and the chicken will come out no feathers, but all scales, okay? Just like a reptile. You can tweak the gene that causes the chicken to form teeth versus just a beak. Tweak the gene that causes a chicken to form a tail. Okay, so all these genetics, we can tweak and move the genes, and we know that these genes must have mutated from reptile to bird, or from dinosaur to bird. Okay? But that evidence is also in the fossil record. We have transition species like Archaeopteryx, which have teeth still on their bill. And they have claws on their front of their wings. They have a tail. They have scales and feathers. And so this shows that transition between dinosaur and bird. Okay, so here's an example. Titanotheres. This is an example that we can use multiple pieces of evidence from the fossil record to distinguish what was going on. Okay. So about 50 million years ago, a Titanotheres would look like this, and say 30 million years ago, it looks like this. So what's going on to make this organism adapt and change to get different forms? Okay? And what we see is when we start looking at the habitat that these organisms lived in, Titanotheres in this 50 million years ago, okay, lived in a forested habitat. It was small, streamlined, it could move between the trees. As the forest in this region decreased and became more savanna and grasslands, and you had less and less trees, the organism itself bulked up and started to produce like facial projections, horns, whatever you want to call them, probably for the purpose of protection, because we find it in both male and female species. And so you can see this transition from forested habitat into a savanna habitat with some forest, into more grasslands with just a few forests, into pure grassland habitat and you get this body shape and body size form change. Right? And that's all visible in the fossil record because it's not just titanotheres that's fossilized, but the plants are fossilized with it. Its fecal material is fossilized, so you can analyze, well, how much grass did it eat, how much trees did it browse on, etc. And you can start to see that. Along with it, though, you can also see predators, right, that have been fossilized, and you can see the differences in predators that you would see in that region. Okay, another source that Darwin had was the anatomical record. So even though Darwin was not a big fan of surgery, and he actually, was fairly kind-hearted and really he enjoyed the company of people, even though he was kind of a hermit and he didn't interact with people very much later on in his life, and that probably was because he was always sick. And he vomited every single day, basically since he got back from the voyage of the Beagle for the rest of his life. So he was always sick. He had bad vertigo. And so he didn't like to go into public and things like that. But he did like people. And that's why medical school really bothered him a lot. It was because of the pain that people went through in order to, have a medical procedure. But what Darwin did take away from med school was this ability to dissect things. Okay? So he didn't like surgery, but he liked dissection. Okay? And he would use dissection to collect a lot of data on things like the anatomical record, but on other things like similarities between species. And so he, had early on dissected out vertebrate embryos, and he would look at the embryos at different stages of maturation, and he noticed that all embryos will have pharyngeal gill pouches. So they have these gill pouches that are present when they're in embryonic form. They'll also have tails, and there's a lot of other things that these organisms had. But Darwin noticed that all vertebrates share the similar set of development features. Okay? He didn't know what we know now, and we know the genes that control for where the head's produced, where's the tail, where's the thorax, these kind of things. We know these genes and we know where they're placed. They're called Hox genes, and it determines where you're going to produce arms and legs and things like that. And that's why we see this similar form between all vertebrate embryos. And we've manipulated, and even invertebrate for that matter, we've manipulated Hox genes and we've moved them around so things like fruit flies can grow a leg out of their head and an antenna where the leg should be, and these different things, because these genes are determining where that structure is placed. Right? And that's why the instructions or features are very similar. So he knew early on that it was very difficult to tell the difference between embryos. Most people cannot tell the difference between an embryo at this stage. One of these embryos is human, and most of you, if you were to guess right now, you would be wrong. But the human embryo is this one. Right? I mean, it has very large pharyngeal gill pouches and a huge tail. Right? But one of the things that gives it away quite a bit is this placental connection. Okay? This is a bird, no placental connection, and this is a reptile, no placental connection. Okay? But nonetheless, you can see the pharyngeal gill pouches on a human much larger and much more distinguished than you would see on a bird or a reptile. Okay? And Darwin noticed this very early on in his gathering of information. The other thing that Darwin noticed was what we call homologous structures. Sorry about that. Homologs, or homologous structures, that will occur when you're using the same bones but for a different function. Okay? So these organisms will have an ancestral connection, and they might be using the same bones. Maybe they're using the humerus, the radius ulna, and the phalanges, okay? But they use them for different purposes. Ours, the purpose, grasping things, tree branches, things like that. But something like a porpoise would be used for like flippers, or a bat would be used to fly. It's the same bone structures, just different function. That's homologous structures. Okay? Analogous structures are structures that look like they should have the same ancestry because they work the same way and they look very similar. Okay? But the linkages between lineages of those organisms are not there. For example, if you look at, say, the wings of a bird and the wing of a bat, the wings of, say, butterflies, okay, or beetles, okay, these all have wings and they all work in the same general functionality, but they're made of very different things. Okay? And so there is no connection between bird wings and bat wings. And those two developed completely separately from each other. And that is what we call convergent evolution. It's a evolutionary design that works. Okay? So through evolution, the practice of being able to have, things hanging off your appendages and flapping your wings to fly probably started with gliding first. That's where most of the evidence suggests. But eventually the gliding system and the flapping system come together and that organism can fly. There are different ways at which, though, those parts come together. So looking at homologous structures, or homologs, okay, you can look at vertebrate limbs, and like I said before, you have a human, so humerus, radius, ulna, phalanges, okay, and you can compare that to a porpoise, same bones, just a little bit different structure. A horse, same bones, just some of them are fused together and some of them are more robust. Okay? Bat, same thing. So these are homologous structures. The bone, it's bone for bone, same bones. It's just the bone itself is either more robust or shrunk down. It might be fused together. So there's a little bit of difference there. Convergent evolution or analogous structures. Okay, like I said before, you have things that can fly. They all have wings. Okay? Now that wing could be what we call a wrist wing, like birds have. So they don't actually have, phalanges. They've all fused together into one, kind of wrist or one ball, and then the feathers are attached to that. You could have what we call arm wings, which, Tesauras and other dinosaurs would have had. And so these are just flaps of skin that would be attached to the arm that they could use. And then you have something like, bats that would have what we call hand wings, where skin is stretched between fingers, and that's the main part of the wing. The other thing that we can see is, with convergent evolution, is examples of groups of organisms that have similar life cycles and lifestyles, but are not related to each other. For example, marsupials and placental mammals, they evolved separately, completely separate. Australia had broken off on its own, and placental mammals developed in like Pangaea and Africa continents, away from Australia, and marsupial mammals developed on Australia. Okay? And yet we still see very similar examples. Okay? We have marsupial mouses and placental mouse. We have the flying flanger, okay, which is our marsupial, okay, and the flying squirrel, which is placental. You have the Tasmanian wolf, which was marsupial, and it is extinct now. And then our gray wolf, which is a placental. Very, very similar habitats, very similar features. They look the same, okay, but they evolved in different ways. They converged on the same path, but they're not related to each other. Okay? And that's where convergent evolution comes in. All right, so other evidence, okay, that Darwin was not aware of comes at the molecular level. Right? And so the discovery of DNA, the discovery of the structure of DNA, the discovery of genes and proteins really has allowed for science to test a lot of Darwin's hypotheses, okay? And a lot of the research that Darwin had suggested was to be true. Darwin made the suggestion in the late 1800s, or I guess 1860s, okay, that humans and chimpanzees were closely related, that they were our closest ancestor, and based on blood chemistry, facial features, okay, overall just general patterns, this was our closest relative. Well, it wasn't until the discovery of DNA and our ability to manipulate DNA and examine DNA through like the Human Genome Project or the Chimpanzee Genome Project or the Gorilla Genome Project, it wasn't until we got to that point where we could actually test, well, is Darwin correct? Are we more closely related to chimpanzees than we are to gorillas or orangutans? Okay? And indeed, it's true. Darwin was right. We are more closely related to chimpanzees than gorillas or orangutans. We share about 98.8% of our DNA with chimpanzees. There's about a 1.2% difference. Okay? And so things that have been separated for a longer period of time should have more differences. Okay? And we can look at that at the protein level, but we could also look at it from the gene level. So if we look at it from the protein level, we can take something as simple as hemoglobin, the protein that allows for us to bind up oxygen, supply cells with oxygen, and bind carbon dioxide and rid our bodies of carbon dioxide. Some of the most ancient organisms on the planet, like lamprey, which are, really kind of the very first vertebrates that we have evidence of, they actually have only 3 vertebrae, okay? And the rest of the organism itself is all cartilage. There's no bone, but it does have 3 vertebrae that attach to the head. It doesn't have a skull, okay? And this is considered an animal, and so we're looking at very prehistoric, ancient animals, lamprey. They have hemoglobin, they have the protein, okay? And if we look at the differences between a lamprey hemoglobin molecule and a human, there's a 125 amino acid difference. As we progress with things that are closer to humans, right, like monkeys, okay, remember, chimpanzees are our closest relative, okay, and chimpanzees and humans have the same hemoglobin molecule. But we just have to diverge a little bit back to monkeys, like macaques, and there's 8 amino acid difference in the entire strain of amino acids. Okay? And like I said before, most proteins are, more than 300 to, 1,000 amino acids long. Okay? And so we're talking not very much of a difference between humans and monkeys, and there is no difference between humans and chimpanzees. Okay? But it really figures out pretty well. Mammals are closer to humans than birds. Birds are closer to humans than frogs. Frogs are closer to humans than fish. Okay? And it really shows this evolutionary divergence of hemoglobin molecules. And we can do this with lots of different proteins. All proteins will show similar traces if they have the capability to have evolved and changed. Okay? Now, some proteins are so specific that they rarely change at all. Right? And you look at the protein in a lamprey and the protein in a human, and they're still identical. We're talking hundreds of million years later, and they're still the same protein. The other thing is we can start looking at evolutionary change or evolutionary mutations because some mutations accumulate at a constant rate. Okay? And it's predictable how many years would you see before you'd see a slight mutation, maybe 1 amino acid difference or something like that. that change in a single gene, well, that allows us for us as scientists to then document, well, when was the last time these two organisms shared the same gene? Okay? By knowing that, we can know when was the last time they diverged from each other. So when did they last share a common ancestor? Okay? And that's what we call molecular clocking. So you can take and you basically do the same thing that we do with radioisotope dating. We get a date. But now we're doing it with genes and evolutionary change of those genes. A good example of this is cytochrome C. Okay? Cytochrome C is a gene that's found in every organism that has an electron transport system, which we know that any organism with a mitochondria will have an electron transport system. Okay? Even organisms without mitochondria still have electron transport systems, but concentrating on organisms with mitochondria, cytochrome C is the protein that allows for oxygen, well, electrons, to bind up to oxygen to produce metabolic water. Okay? And we know by looking at a protein that is, as important as cytochrome C, organisms can't live without this. Okay? But nonetheless, we can examine it and see that it has changed, a little bit of tweaks for a long period of time. Okay? But these tweaks, these changes, occur at a fairly constant rate. Okay? So we can examine this when we're just looking at, say, mammals. And we can say, all right, we can take those Australian mammals, okay, that are going to be marsupials, and you can take, non-evolved Australian mammals or placental mammals like a human, and you can see when was the last time these two organisms shared a common ancestor, or when was the last time they shared the same cytochrome C gene. Okay? And it was roughly 120, 125 million years ago. The nice thing about being able to do and utilize molecular clocking is we can then say, all right, based on this, can we then correspond that to the geological record? Okay? When did Australia break free from Pangaea, from Gondwana, from the main continent? Well, that's about 150, 130 million years ago, based on geological evidence, is when it broke away. So that's fairly fitting, in that humans and kangaroos shared, that ancestral cytochrome C about 125, 120 million years ago. Now, realize that humans are not 120 million years old, and neither are kangaroo. So this is an ancestor that would have been shared between these two groups that would have this cytochrome C. Okay? And if we can just fast forward all the way to modern day, and we know that you can mate a horse and a donkey together. You don't get a viable offspring, you get a mule. It's sterile. So that means that the horse and the donkey are separate species. That's one of the key components to the biological species concept, is that if you mate the organisms together, they do not produce viable offspring. Okay? That makes them two separate species. So horses and donkeys are separate species, yet their cytochrome C gene has only been separated for about 1 million years. Okay? So they're very closely related, and this allows for us to look at how close organisms are related. So that kind of brings me to some of the criticisms to evolution. So evolution, more than any other theory in biology, has critics and criticisms for that evolution. And mainly it's because it doesn't follow, evolution doesn't follow most of the religious doctrines as they're written. Okay? It doesn't mean that you can't interpret the religious doctrines differently, but as they're written, evolution does not follow those doctrines. So people have criticisms when it comes to evolution. Okay? And it can be very bitter, and it can be very emotional, and can cause a lot of controversy in public. It can be found in school boards, it can be found in city documents, it can be found on college campuses, it can be found in elementary schools, high schools, all over the place. You can find examples of this controversy between evolution and religion. And it really started with the origin of species, right? But it still occurs today. And with new groups popping up, like intelligent design groups and things like that, they have different criticisms towards evolution. So the main criticisms, or main groups of individuals that have criticism of evolution, would be the fundamentalist movement, scientific creation movement. Local action would come from school boards or other entities that might have a say in what children are exposed to, things like that. And then the most recent would be what we call the ID group, or the intelligent design group. Okay. Now, what are their criticisms? Their criticisms mainly are these 6 things: that evolution is not solidly demonstrated, okay? Although I will show you some examples where there's quite a bit of evidence and we can demonstrate evolution in a human's lifetime. Now remember, evolution is change in genes or genetics of a population over time. Now if you're wanting to see an elephant evolve, it's going to take a lot longer than a human's lifetime, because elephants give birth to a calf about 1 in every 20 years or so. Okay? I mean, it takes them longer than 20 years to mature, and then they don't have very many calves. So maybe in a human's lifetime, a single female might produce four babies. That's going to be very difficult for you to see evolutionary change. But if we take bacteria, which can reproduce itself and again and again and again, and you might get 1,000 copies of bacteria in a day, that's a lot easier to demonstrate evolution. And there's lots of documents showing this. The intelligent design argument suggests that there are features that occur in humans that only an intelligent designer could make. So something like the eye, the human eye. No fossil intermediates, okay, and I'll show you that indeed we have lots of fossil intermediates. That evolution violates the second law of thermodynamics, which is that entropy increases over time. So disorder increases, but evolution actually suggests that it's not, that things are not getting more disordered, but rather they're becoming more orderly. Okay? And the problem with that violation is really the second law of thermodynamics, or entropy increases. The piece that a lot of individuals who are criticizing evolution for, the piece that they're forgetting is the second law of thermodynamics only works if energy is eliminated from the system. So if you eliminate energy from the system, then entropy does increase. But there's no evolutionary system, there's no biological organism that can have energy eliminated from the system and still be alive. Now you have to constantly supply energy in order to maintain cells and normal functions like growth and reproduction and normal maintenance stuff. You have to supply energy. And that's why evolution does not violate the second law of thermodynamics, is because energy is always supplied. And so it doesn't really apply to the second law of thermodynamics. Natural selection doesn't imply evolution. This is absolutely true, and most biologists would say so, because natural selection is nature selecting an organism to survive. If the traits that allowed for that organism to survive are not passed on to the next generation, then there's no evolution. The genes in the population are not changing. And life could not have evolved in water. This really has nothing to do with evolution. It has to do with how life came to be. So it really doesn't address evolution at all. So most of these objections, okay, have been refuted numerous times. And then in other cases, they have nothing to do with evolution, and it's just a way to rant or criticize Darwin's theory of evolution by means of natural selection. So some of them that we can demonstrate fairly easily is the human eye. Okay, so the thought that the human eye is so complex and so perfectly designed that there had to be a designer, well, we can look at the flaws of the human eye. Okay. First of all, the human eye, the pigments, the receptor cells, so the cells that are picking up photons and producing an image, occur behind nerve cells and blood cells. The problem with this is when your blood flow is increasing a lot, right? So let's say you go out and you run, and you're playing basketball or something, or soccer or something like that, and you're running a lot, and then you sit down, and you cut off the blood flow by sitting down, and then you stand up, and all of a sudden you see stars and dots and things like that. That's because the light has to travel through your nerves and your blood vessels to get to the receptor cells. This is a horrible design. Okay? And invertebrates, okay, like the octopus, which has basically the same eye design as we do, can see the same visual cues as us, same color patterns, basic same eyesight. Their design is better than ours. Their nerves, blood vessels, etc., all run behind the receptor cells. So they never experience those light blurs and things like that. So the vertebrate eye is not very well designed, first of all. And then apart from that, we know that you can have intermediates of this. We know that you can remove or change the amount of material in front of the eye. So the actual lens of the eye, you can change and alter, the angle, or alter, how deep it is, and that'll give you a different vision, or to give you a different image. And lots of organisms have varieties to this. So the eye works in many different forms and is not very well designed. At least the human eye is not. No intermediate fossils. This fossil here is Tiktaalik. Tiktaalik is half amphibian, half reptile, or half amphibian, half fish. If you look at it, it has scales like a fish. It has fins like a fish, but if you look at where the wrist would be, it is bone for bone, the same kind of wrist bones that you would find in vertebrates that can stand. The other thing is if you look at the eyes are on top of the head, suggesting that this organism was going to the surface to take breaths of air. We also have fish that have lungs, like lungfish, that would be an intermediate also, that are breathing air from the surface and not using gills. And there's many cases in which we have intermediate fossils. Archaeopteryx, you have Tiktaalik. We have living creatures that aren't fossils but are intermediates, like the platypus, which is intermediate between a reptile and a mammal. Still produce eggs. Okay, they don't have nipples, but they do have mammary glands, and so there's a lot of these transitions into what we would call mammal from the reptile point of view. Okay, so with that, we're going to move and we're going to talk about population genetics. So we'll talk about Hardy-Weinberg and how we calculate whether or not a population is actually evolving. There's a mathematical formula in which we can sample a population and then suggest whether or not it's deviating from what we would normally expect. And when we say normally expect, we're talking about the ratios of homozygote, dominant, heterozygote, homozygote, recessive. So normal Mendelian genetics comes into play again when we're trying to figure out if a population is truly deviating from normal Mendelian numbers. Okay? That would suggest the population is evolving. It's changing. The genetics of the population are changing. Okay? All right, so next time we'll talk about that.